This lesson outlines cloning an animal by transplanting a cell nucleus, cloning an animal by transplanting embryos, and the meaning of a transgenic organism. Cloning by somatic cell nuclear transfer involves placing the nucleus from a donor body cell into an unfertilized egg after the egg's own nucleus has been removed. First, a donor animal is chosen, and a somatic cell, such as a skin cell, is collected and its nucleus is isolated. Second, an unfertilized egg cell is taken from another animal, and the egg nucleus is removed in a step called enucleation. Third, the donor nucleus is inserted into the enucleated egg, and the cell is activated with a brief electrical or chemical pulse so it behaves like a zygote. Fourth, the activated cell divides in a culture dish until it forms an early embryo such as a blastocyst. Fifth, the embryo is transferred into the uterus of a surrogate mother, and the offspring that develops is genetically identical to the nuclear donor and not identical to the egg donor or the surrogate. Cloning by transplanting embryos can be done by embryo splitting, which means dividing a very early embryo produced by normal fertilization into two or more embryos that can each develop. Each split embryo is placed in the uterus of a surrogate mother so several identical offspring can be born, and those clones are genetically identical to one another. A transgenic organism is an animal whose genome contains an added gene from another organism or a modified version of its own gene that is inserted in an early embryo so the new gene is present in the cells, is expressed as a trait, and can be inherited by the next generation.